Hello everyone, and welcome to this week's Biology 1124 lab. I'm Shannon Reeves. You should be in section 30 or 40 of the online lab um, assignments or sections in that group. So if you're listening to this, just make sure which section you're in because you'll be required to post Flipgrid videos according to that section. Alright, so I'm not doing a video today. I'm not feeling really great, and so I figure the only signs that you need of me being sick is my raspy voice. So today I'll just be doing a voiceover, but of course with the help of Miss DJ Ravioli. So this week's investigation, excuse me, has to do with macromolecules. We're going to be identifying and analyzing substances that have these macromolecules. So you will need to be doing these things during lab. You're going to have activities that identify and describe various types of macromolecules. In your lab activity, with the data that we give you, you'll determine the macromolecules in common foods. And then you yourself are going to write up an experimental summary. So keep in mind, this is your first summary that you're going to be making. We have lots of resources on Canvas for you underneath the lab resources and manual module that help you with knowing how to write a lab summary. And we also have examples of lab summaries available for you on Canvas. Then the last thing that will be required of you is to make your first official Flipgrid video that is worth points. So this is a part of your lab summary grade, and you will be provided with the links in the modules for this week. All right, so let's review really quick on the different types of macromolecules that we do have. So first we have our lipids. Lipids, I made sure to add the picture that has the hydrophilic head and hydrophobic tail, but we have this other picture of it as a phospholipid bilayer because we know that lipids in its true monomer form already has two parts to it. But because of this, make it a really good kind of a layer that we can keep things in and out. Next, we have our amino acids, which have 4 groups. So you always have in this blue part right here your essential carbon atom. You'll have your carboxyl group, which has the OH over here in the yellow. You have your lonely hydrogen right down here on the bottom. You'll have an R group, which this determines the shape of your amino acid. And then right here we have our amino group right here. So our next one is carbohydrates. These are 2 carbohydrates that are connected right here. And lastly, we have nucleic acids. So in this lab, we won't be dealing with nucleic acids. We're just going to be dealing with lipids, amino acids, and carbohydrates. It's following the same path as lecture. As you know, we really haven't been focusing that much on nucleic acids, but you will once we get in later into the semester and you start talking about DNA and RNA. So let's go ahead and look at our first group of carbohydrates. So the monomers of carbohydrates are called monosaccharides. We have our monosaccharides right here. They're different types of monosaccharides. And the polymers are called polysaccharides, or in this case, disaccharides. We have versions of disaccharides right here. Dehydration synthesis is how you build your monomers. So if you have 2 monomers that form a covalent bond, that will release water in the process, and remember it also takes energy, but that right here is dehydration synthesis where you're building your monomers together, whereas hydrolysis is pointing to over here where we're separating, and because of that we turn polymers into monomers. We break them down into monomers with the addition of water, so this usually releases energy. So these carbohydrates are a very important source of energy that we get from food, and you'll be observing an experiment where we will add Benedict's reagent to different food substances, and you will need to see if that substance turns orange or red or not. If it does, then it has carbohydrates in them. I would pause this video right now if you have not read your background and done the steps prior to watching this PowerPoint. If you have not done that, I strongly urge you to. This all will make much more sense if you've looked at the background information. So moving on, we have another type of carbohydrate that are starches. So now we're thinking about starches, which are long strings of glucose molecules stuck together. These are in your like potatoes and carrots and cereal and pasta. Many plants use this form of carbohydrates as an energy or for storage. And whenever you do your experiment observation today, iodine will be the reagent that we introduce to these food substances. If it has iodine and it turns black, then it indicates the presence of a starch in that food. So next we have our proteins. Excuse me. The monomer of proteins is your amino acid, which combine together to make proteins, as we discussed earlier. The shape of the protein is crucial to its function, so when we talk about secondary, tertiary, and quaternary structure of proteins, these are important shapes that determine what they'll be doing in your body. So proteins have many functions. First, enzymes, which serve to control the rate at which chemical reactions happen in your body. So they're basically making sure everything is going at the speed it needs to be going. Proteins also have a big role in cell structure and cell transport. So as we know with facilitated diffusion, we have transport proteins. And those transport proteins help push things in and out of the cell that normally are too big or can't go through on our own. As we know with our phospholipid bilayer, it only allowed certain things through. So in our experiment today, the Biuret reagent will show whether there is protein. It will turn purple if the food substance has protein in it. All right, and last up are our lipids. So here again is our hydrophilic head, which is polar, and we know our hydrophobic tail, which is nonpolar. So this means that lipids, when strung together, make like really good compartments. It makes a really good cell wall, sort of like a Ziploc bag. It keeps things in and out, and each of your cells kind of acts like a little Ziploc bag, but you can open that bag and let things in, but only if you have the right equipment, right? So we use a lot of lipids for energy storage and for membranes, cell membranes like we've been talking about. And Sudan 4 is the reagent that we'll be using, which is indicated by a bright red if that food substance has lipids. Now keep in mind that our very first reagent was for carbohydrates for sugars, and it turns orangish red. So keep in mind that when looking at your data that you'll want to identify that those are just a little bit different from each other. So what will you need to do during your lab? We're going to give you the experiment and how it turned out, essentially your data, but we'll also give you some pictures and some information that will help you as if you were doing the lab yourself. So you'll need to identify and describe the various types of macromolecules. You will need to determine the macromolecules in common foods, and then write a summary and comment with your Flipgrid video. So what all does that entail? So keep in mind that we'll have control and experimental groups. We've never talked about positive control groups and negative control groups necessarily. A positive means that it contains the molecule of interest, or only the molecule of interest in our instance. And a negative means it does not contain any of the molecules. So the reason for doing this is that with a positive control, you want to see what a reaction in that, let's say we're testing our lipids, and we have that Sudan 4 reagent. We want to look and see what it will look like to actually turn that bright red, so that later, whenever we have an experimental group and we're not sure whether it's supposed to turn bright red or nothing at all, we will know what that bright red looks like. So this positive control is essentially whenever we have something that reacts the way that we intend on it reacting in that situation. if that molecule of interest is present. A negative control does not contain any of it. So a negative control is not going to turn red. So that way we know exactly what our experimental groups look like if they do not react to that agent. So we have 4 experimental groups. That means you have 6 groups total. This is not like what we did last week where we have 1 that is a control and 1 that's an experimental, or we had 2 experimental. This is more like a new type of experiment that we haven't done before with 4 different experimental groups. All could contain any of what we have or none at all. And then we have 2 controls, 1 positive that does react and 1 negative that doesn't react. And we'll go over what positive and negative controls are just a little bit more in an experiment or in an example up ahead. Keep in mind that we also have dependent variables, which are the type of macromolecule present, and independent variable is the type of food that is tested. So we're giving you your independent and dependent variable. This is a small President's Day gift from Dr. Shaw, because those are definitely points on your summary, so hopefully you don't miss those points. Oh, ravioli. We did need a ravioli break. Okay, so let's talk a little bit about positive and negative controls. So, ravioli lives for food and the occasional pet from me, although I'm pretty sure she could live without it, but she cannot live without food. And recently we tried to switch her dry cat food to something more healthy, because ravioli likes garbage food, basically the kind that has like virtually no protein and has all the horrible yummy preservatives that make it taste like it could be real food, although it's definitely not real food. The kind that the FDA is even like, hey, don't feed this to anything that's living because it's not food. But she loves it. So you may recognize Temptations. They are sold like everywhere, even at like not, you can like go to the gas station and you can go to a tire store, and I'm pretty sure they're everywhere. They're impossible not to find. But that's because they're garbage. So she likes them more than she likes basically anything. And so I have actually a picture of the label right here, since we're talking about food and what food's made up of. The label here is 28% crude protein. So basically everything else, 72% of this is basically garbage. They're made by Mars. I don't know if you remember this name. You probably do, but Mars makes junk food for people too. So of course, why wouldn't they make garbage food for pets also? So we don't want ravioli to be a garbage kitty. So we thought maybe we can find another food that is more healthy that at least she'll tolerate. We've tried this in the past and we got these things, wellness treats from Petco or PetSmart, some cat or dog place, I don't know. And we thought that she would at least tolerate them, and she proved us all wrong, and she just really hates anything healthy. So we're going to try this again though, because we're determined to make her a healthy cat. So we found another set of treats that was recommended called Whole Life. I don't know. I probably wasted my money again, but we'll see. So new treats, this is our experimental group, because we don't know whether it's going to be a good reaction from ravioli or a bad one, right? Temptations, though, that's our positive control. We know we get the reaction out of ravioli that we want, which is for her to eat that stuff. Wellness treats, we know, serve as our negative control. This is negative ravioli. She doesn't want to eat the food. She's still hungry, and it's your fault. So, as you can see in that scenario, we have those positive and control groups. The positive being the temptations. That is a reaction that we want to get. So I'm just repeating this slide again so that now that we've learned a little bit in another example about what control groups are, and maybe revisiting it is a little bit more clear. But control groups are where the treatment is essentially is going to be positive, it's there, the molecule of interest, or negative, the molecule of interest is not there. Now, in our previous experiment, we weren't worried about a molecule. We were worried about a reaction from ravioli. And we got either a good reaction, she eats the food, or a negative reaction. So that's why we're changing necessarily what the positive and negative control stand for. But we still have our experimental groups and, again, our independent and dependent variables. So what was the conclusion, though, to ravioli? Ravioli loves garbage food. She's never going to change. Accept her for what she is, please. So for 1 week with each of the treats, we gave her those treats only. So we have over in our legend our new treats. So out of 7 days, how many days did Ravioli eat that treat? 1 day out of 7. She would rather starve than eat healthy food. The next one is temptations. How many days out of the week was she willing to eat that on that week? All 7. She loves garbage, that's why. And the last one, wellness treats. She ate them 3 out of 7 days. So my conclusion to this would be maybe if we kind of mixed our 2 treats, she'd be more willing to eat healthy food, but I'm not going to hold my breath for Ravioli's palate to change anytime soon. So hopefully that gives you a little bit of idea of control and negative groups. So remember that you need to write hypotheses for this this lab, and it's actually a little bit different than last lab. So down here on the bottom it says we will be writing a hypothesis for each of the 4 foods being tested. So remember, our 4 foods are hot dog, tofu, energy drink, and milk. So you need to write a hypothesis about each of them. Here's an example. Red Bull turns black in the presence of iodine because it contains starch. You can use that one or not. But notice that it has because or a causal statement in it. But the goal of a hypothesis is to be testable. It doesn't necessarily have to be supported. Now remember, hypotheses are not true or untrue. They're either supported or unsupported. So this hypothesis, you don't have to support it in anything. You just need to be able to test it. So in each of the tubes, you have 6 tubes, one for your control, positive control, negative control, and then the 4 experimental groups. They will be labeled for you. You'll be having the 4 different reagents to identify specific macromolecules. So we'll do 6 tubes, all with iodine. Then we wash out all those tubes and we do 6 tubes with the Sudan, all the same foods again, and your control and your positive control and negative control. Then we do 6 tubes of the Biuret and then 6 tubes of the Benedicts. So you will have a lot of tubes to be looking at this week. So these are tips for whenever you're actually in lab. Remember that whenever you're handling these things, you want to use gloves and goggles. Fortunately, this week you don't have to do any of that, but keep that in mind for the future. These details might want to be known for the background information that you'll be writing in your summary. So we are going to be transferring 2 milliliters of the test solution into each tube, and you'll add the amount of reagent that the protocol asks for. We'll be plugging in a hot plate for the Benedict's test because the water needs to be boiling in order for that one to show up. So as I said, we're going to be boiling stuff. Be safe, don't burn yourself. Use test tubes whenever you're doing lab, not virtually. And make sure to clean up after you do your fake experiment online. Alright, so whenever you have finished everything in class, and this is a good reminder for anybody that does go to in-class stuff in the future, always be a good laboratory participant and clean up after yourself. So this is the 3rd time you should be seeing this slide. Hopefully you understand that you're going to be going through your data that we give you, recognizing and determining which macromolecules are in common foods, and writing an experiment or a summary and commenting on Flipgrid about what you will be doing. So just a reminder about displaying your data. When you have finished collecting your data, summarize all that you have recorded into one table. So keep in mind though, this is the only table you will ever use in this class. You will not use a table instead of a graph in any other summary. If you do, you get 0 points for that because it's not a graph. You would have submitted a table. But in this particular experiment, yes, you will be doing a table. The table should contain information about which molecules are present in each food sample. You'll place the independent variable in the leftmost column, like table 2 in the lab background, and then place the dependent variables across the top row. If you have any questions about how to make graphs or tables in Excel, of course, feel free to email me or to reach out and we can meet up in office hours or over Zoom, or I can just shoot you an email back, whatever you prefer. The last part that you'll be doing as a part of your summary is finding and citing scholarly sources. So last week we learned how to use the library database to find and cite scholarly sources. This week we have to do it for 2 specific articles. So you have to use the same place that you went to before, Web of Science, and you'll be searching for the database using the terms macromolecules as the topic. So feel free to come back to this portion of the slideshow as you cite or you find your citations. So you might want to make sure and pause this and write this down if you would like to as well. But you will be asked to correctly cite 2 journal articles from the list in the literature cited section of your lab summary. So they are the first 2 that I want you to cite. Just make sure they are the top 2 articles. If you do article number 2 and 3, you will get half credit. If you do article 3 and 4, you will get no credit. But you will do this at the end of your summary, and you will create a literature cited section at the end of your lab summary as well. You do not need to use the paraphrase in your actual assignment. We will work on this next week, but you need to be able to actually cite properly your 2 sources. Okay, so don't forget, review the how to write a lab summary document and review the assignment rubric before writing. The lab summary should be uploaded to Canvas this Friday by 5 p.m. as always. And here are the following things that we would expect you to address in your Flipgrid video. Remember that I'm available all week long. I'm here during office hours. I am here over email, or we can set up a time to meet either online or over Zoom. But try and not keep this into the last day. I typically don't answer emails after around Friday at 4 or 3:30 p.m. So if you have good questions about the lab summary, about what you have to do this week, or just general help for lab or lecture, feel free to reach out. You have my email at the beginning of this PowerPoint, and good luck this week.